I mean, those of you who want can make a note of my email ID, so you can chat with me anytime on my email. Now, what are the things I'm going to speak is the biology of the virus, about the disease, symptoms, diagnosis, treatment, prophylaxis and vaccines, types of vaccines, and immunity, basis of immunology. So now let me go into the basis of coronavirus itself. Coronavirus is an RNA virus. It's the largest RNA virus that we have known as of today. It has a length of about 125 nanometers and 330,000 bases. I'm not saying base pairs, remember. This is because it's a single strand. There is no pair. It is a stretch of 30,000 nucleotides that are chained together. So the coronavirus is the largest viral genome, and it's almost 3 times as big as HIV and twice as much of the influenza virus. This virus is grouped into 4 categories or 4 genera: alpha, beta, delta, and gamma coronavirus. This is a class by itself and not to be confused with the variants of the virus that's coming and is constantly bombarded on the newspaper and the news channels that there's an alpha variant, beta variant. Those variants have nothing to do with these 4 genera over here. All of them are the single class of coronavirus that we are speaking here. There's 7 known types of this virus, and we are all speaking about the alpha coronavirus that is the causative of this huge pandemic, wherein all of us are going through so much of trauma, as a physical, mental, psychological, and everything else. Um, so this is how the virus looks. The main things is, I mean, I would like to tell you that you should remember how many code proteins that these have, or how many major proteins it has, because at a later point when I'm speaking about vaccines, I would again refer back to these proteins that are mentioned here. The most important is the S, or the spike protein, which is a glycoprotein in nature. Then we have an envelope protein. Okay, centrally is the RNA, which is 30,000 bases long. This is covered by a nucleoprotein, which is again sheathed with an envelope protein and then a membrane protein. And on this envelope, we have embedded the S-glycoprotein, which is the spike protein, which everyone has been hearing constantly. So now let us see how this virus creates all this havoc. So there is, in every cell of our body, every cell, we have what is called an ACE2 receptor. So this ACE2 receptor, if this is a human cell that we have over here, the ACE2 receptors are present all over our cells in the body. Now I'm taking only one site here for the sake of clarity. So this receptor protein on our cell opens up, and this virus over here comes and with the help of the spike protein attaches to the ACE receptor here. And there are other functions of these proteins right here, which enables us for this to fuse in here and enter into the cell. Once it's entered the cell, then what happens is it releases its RNA. Now, all viruses, the most important thing of viruses, it's a total parasite. It can't do anything by itself. If this virus particle were on the table, chair, or book or pen, it is as good as a dead thing. It's like dust particle. It cannot do anything by itself. It's only when this virus or any virus enters a living cell that it becomes a living organism and starts expressing its characters. So once it enters the cell, it releases its nucleic acid. This nucleic acid, the viral nucleic acid, uses the cellular machinery, that is the whole cell machinery, the ribosome. It makes first its viral replicase protein, which enables the viral RNA to be replicated. Now, if you recall the first slide, I said it's a positive stranded virus. So, what is positive? Positive means this strand can directly code for the protein. Now, if this were to replicate, it has to go into a temporary stage where it will copy this into a negative strand, and that negative strand will again make a positive strand, multiple positive strands, which again are embedded or packaged into the virus. So the virus replication is very specific in the fact that it's like an assembly line. It's like an assembly line. The virus enters the cell, it releases its RNA. Once the RNA is released, then it makes all its daughter RNAs, and on the one side it's making the daughter RNAs, and on the one side it's making the proteins that it requires. And once these proteins are synthesized, then it packages the virus into the protein that it is made, which is assembled to form a neat shell like this manner. So this packaging is done, and this packaged virus is now budded off. So that essentially is the life cycle of a typical virus. So today you're hearing that this spike protein is mutating constantly, and we have today what are called as alpha variant, beta variant, and a number of other variants. So what is happening is it is mutating. Now, why is it mutating? You know, we have this whole story like I punch you, you punch me back. Or to put it in Hindi, we would say nehle pe dehle. That is, if the virus, we are trying to play with the virus and see that it does not disable us, the virus is trying to see that it is disabling the property of which we are disabling. Therefore, when the virus mutates, it has a lot of advantages that it confers on the virus per se. What is the advantage? It enables the virus to sidestep all the vaccine that we are making. Then, not only that, these viruses, I mean, there is no hard and fast rule that only one virus should infect you. Suppose a cell is infected by one or more kinds of viruses, they recombine or they swap chunks of their RNA, which gets packed into its own shell. And this kind of gives the virus a very deadly dynamism. So originally, in bats, which carry about 61 known viruses which carry infect human cells, so these harbor as many as 12 variants within them. But these 61 viruses do not harm the bats. But the theory is the immune, like how it doesn't affect the bat is it has a unique immune system. But I shall not go into the bat immune system. We will stick to what we need to do for humans. Now, what is a mutation? A mutation is a heritable alteration in the nucleotide sequence of a genome of the organism, be it virus or man. So when such a change occurs, these mutations, based on the kind of function that they do, can be broadly classified into 5 types, which are neomorph. Neomorphs are those that do a novel function, which is in comparison to the existing function. We have what are called hypermorph mutations, which are hyperactive compared to the existing mutant. For example, today we hear that, you know, one particular variant is more infectious than the other virus strain which exists. So that is a hypermorph, and the original one was a hypomorph in comparison. Then we have so many mutations which are amorph. It does not bring any change at all. Whether it's there or not there, we don't even perceive it. So such mutations are called amorphs. And then we have mutations which are called antimorphs, which are antagonistic to the original. So these are the 5 kinds of mutations that can be broadly classified, not only in terms of coronavirus, but in terms of any mutation that occurs across biology. So when these viruses replicate, they do not exactly replicate themselves, but then they introduce this variant nucleotide into its daughter cell, and therefore some of them establish or themselves, while some of them die out. So the more effective ones, effective for the virus, evolution is always in terms of it improves that particular species. It does not look to improve me. It looks to improve its own self. So those mutations that occur in coronavirus, which is helpful to the coronavirus, will be fixed in the evolution, whereas the ones which are not useful to it over time die out and do not survive. Now, so the recently identified mutations in coronavirus are alpha, beta, delta, gamma, lambda, and who knows, will go up to zeta, I don't know. But essentially, these are the mutations that are known. So these mutations are because of certain substitutions or combinations of substitutions in the spike protein. And these mutations, the spike protein, impact upon the receptor binding capability. In the first slide, I showed you that the ACE receptor, when the virus comes in, there is a binding between, there is a handshake between the ACE receptor and the spike protein. So this variations that are occurring due to mutations can make this handshake stronger than what it was, or weaker than what it was, or not handshake at all. So these are the things, but what concerns us are those mutations or substitutions or variations thereof, which enables a strong binding to the receptor binding site. It reduces the neutralization antibodies. Therefore, when our body is producing any antibody, it is not as efficacious, or it is playing havoc with the treatments that we are doing. So these are the specific markers with which these mutations will impact upon, and based on how many of these characters it is trying to override is going to be its fate in the biology. Now, let us see, where does this virus come from? To speak about the origin of the virus, one speaks about many controversies over here, and I'm not going into delve into the controversies, both because of the political, social, and scientific agendas that each one of them carry. But nevertheless, I would like to make a point that the most convincing argument that is found that enables us to believe that it is a lab virus is because the spike protein, which indeed binds with the receptor binding domain of the cell, is not found in the bat protein at all. This single character is the one which has made this coronavirus. Either for some reason it is mutated, we don't know how it mutated, but nevertheless, this is the reason where one thinks that this area was played with in the lab, and for some reason it leaked, or whatever happened. And today, because of this variation or this spike protein, which is able to adhere or shake hands with all the ACE receptors present in the different cells in the body, is what is causing havoc. And this is the main reason why people are getting infected. Essentially, viruses also have another property wherein we call it tissuetropism. That means a respiratory virus remains a respiratory virus and does not become an alimentary tract virus or a neural virus or neurotropic virus and other things. But unfortunately, for coronavirus, because it is adhering to the ACE receptor, and all the cells in the body have an ACE receptor, it makes no bones about which system it's infecting. So it has crossed the tissue barrier, and it goes into not only the respiratory tract, but all other cells of the body. Now, how in the world does it enter us? The virus is spread by the droplet infection, and these droplets could come out in terms of your sputum, that when you're talking it comes out, or in your cough or whatever. So normally, if you're exposed to a few particles, nothing happens. But then if there are way too many particles that is affecting us, then it goes all the way down to the lungs and causes infection. So, we have many respiratory viruses, and like many other respiratory viruses, its biggest target is the lungs. So when coronavirus, sarco, and influenza infect and go down to the lungs, it destroys what is called an alveoli system, which is present in your lungs. So when these tiny sacs in the lungs are the ones that shuttle oxygen from what we are breathing into the bloodstream, enabling oxygen presence in the body. And when this sac is full, the virus breaks the alveolar sacs, and it leaks the fluid into it, and the blood also oozes out. This is the reason why clots may be formed, and if the blood is not cleared immediately, it forms a clot and is stationary there, it leads to thrombosis. And this thrombosis is because of which we are reading so many acute cases of the coronavirus where people are dying, and this thrombus or this clot heads to the brain or to a very important location in the heart or wherever. That is called thrombosis. When this clot moves in the bloodstream and lodges itself in different positions or important charaha in the circulatory system or in the brain or very leading points, it causes very dangerous disease. And therefore, we are seeing all these nervous systems, cardiovascular systems where being affected and people dying. So the other cells, including the white blood cells, plug up the airway further, and this causes your breathlessness, the gasping, etc. And it can also, um, the first thing that the virus, when it enters the cell body, our immune system is activated. And when the immune system is activated, we have what is called an cytokine storm, which I'll come to later when I'm speaking about the immunology. But this cytokine system further damages the tissue and makes the disease go worse, and there's inflammation, tissue damage, etc., leaving the person never to recover or with scarred lungs. So as I've already said, because the ACE receptor is present in every kind of cell, it can infect all cells and cause various tissue damage. So most people infected with the COVID virus will have either mild illness or a severe illness. And if they're already older people and having comorbidities like diabetes, chronic respiratory diseases like asthma, or they have cancer and other serious illnesses, this again is going to impact more severely on the patient. So the best way to prevent or slow down the transmission is to be well informed about the COVID virus. And that is what we are attempting to do today, and see how well we can do this. The primary spread, as I said, is because of droplets of saliva or discharge of the nose. We have to practice respiratory hygiene or etiquette, wherein you either cough into your elbow or you use a paper towel or a napkin or whatever. So this is how you're going to prevent. Now we'll dwell a little about the prevention and mitigating of the COVID-19 spread. So essentially, one, we all work remote working. Instead of me coming to Bombay and interacting with all of you, I'm doing a remote conversation with you. And this remote working sees that we are all not assembled in a hall, and neither any one of us is spreading any virus, sputum, or anything. So respiratory hygiene, social distancing, hand wash, and face mask, which time and again has been bombarded by all the media, we are all aware. So what do you do if you're going to your workplace? These are the precautions you need to take. Your hand hygiene, even in the site of work, there should be a physical distancing, cleaning, disinfection. You should wear PPE, your ventilation, routine surveillance, rotation in the institute itself. By shift, some people come, some people go, so that you decrease the density, and everyone in the organization is well informed, and there is a routine screening so that everyone is taken care of. The symptoms are again here: fever, dry cough, tiredness, common symptoms, aches, pain, sore throat, diarrhea, conjunctivitis, headache, loss of taste, smell, and you call it, they say it is there. So, by and large, certain symptoms are very well known, like dry cough, fever, pains, etc., and conjunctivitis or diarrhea is less common. Now, the medical condition, there's a whole big list here, which the presentation is shared. You all can look into it. Now, what are the COVID diagnostic tests that are common? We have the tests available in the market are three main types. One is you're testing the nucleic acid, that is the RNA of the virus, or you're testing the antigen of the virus, that is the spike protein, essentially, but even other proteins. Then the serological test is the antibodies that are present in the human. Okay, these so you're testing the antibodies of the protein in the serum of the patient, and the nucleic acid and the antigen of the virus in the human body. So each type of test has its own special features and uses. As the virus infection progresses, we need to have a different diagnostic solution. While the PCR and antigen tests are useful for detecting the virus in the initial stage of infection, and the serological test or the antibody tests are done much later in the infection to see how well we have recovered. So, nucleic acid test detects the viral RNA, the antigen test detects the viral protein, and this detects the patient antibody. So, what is RT-PCR? I think RT over here is a dual extension of the word. One is it's real-time reverse transcriptase test. Oftentimes people speak of only the real-time, and sometimes they refer to reverse transcriptase. All the PCR tests can detect only DNA, and therefore it is very essential that the virus is reverse transcribed. Mane, the virus is an RNA virus. You're reverse transcribing it into DNA, and this DNA is what you're doing PCR. PCR is a polymerase chain reaction wherein one copy of your DNA is amplified to thousands and thousands, 10 to the power of 6 times, where you can see. This is the test which is recommended by WHO, and everyone else follows. And the material taken for this is nasopharyngeal, throat or nasal swab, sputum or respiratory tract aspirates, bronchoalveolar lavage, nasopharyngeal wash, nasal aspirate, etc. And all these are collected from suspected patients. Now, when we're speaking of RT-PCR, everybody comes with a question as what is the CT value? CT value is a coefficient of transcription. So what happens here, if there's one particle that doubles, it takes some time. So in a lesser time, if you have a higher density of the genome, that means that to begin with you had 100, and in 20 minutes has become 20 times 10 to the 6. So 20 into 10 is a very cognizable number. But if you had only 2 viral particles, and that in 20 minutes will not become 10 to the 6. So your CT value becomes higher. So the lower the CT value, the more highly infected you are, and the more the CT value, the less infected you are. So now, having said that, This is the material which we use for the COVID virus. And essentially, in a PCR test, you have the sample from the patient, the primers, and the other necessary elements of transcription first, and then for synthesis of the polymerase to make more and more copies of the RNA. And you see, basically, you get a curve wherein you can see the CD value and the density of the viral particles. So so who are the patients? Whoever is associated with people who are already having COVID, or if they have traveled to a geographical site where COVID is present, etc. So in an antigen test, what you do is, here again, you take the antigen, the nucleocapsid protein or the spike protein is used to detect whether it's also called rapid antigen because you do it on a filter paper. You can also do it at the physician or point of care, as we call it. Like when you go to a clinic, right there, if I can use a filter paper, take a drop of blood and do the test and tell you whether you're positive or negative, that is called a point of care. So what is done in this procedure is the filter papers, a special kind of filter paper, are embedded with the antigen of the virus. And when the patient blood is taken, you put a drop of blood. Hopefully, you have the antibody in your serum and it reacts and gives a color reaction. Or it can be done the other way too. You can have antibodies embedded onto the filter paper, and you put antigen from the patient sample to do a rapid antigen test. So this exactly is the figure I want you to remember, even for a later time when I'm speaking about vaccines. The main proteins over here are the glycoprotein or the spike protein, the hemagglutinin, the envelope, and the membrane protein and nucleocapsid. This is the RNA, which is not will not have any role in the immunology of the virus. So to speak, this spike protein is very important. It contains two subunits, one called S1 and S2. The S1 contains a receptor binding domain, which is responsible for recognizing the ACE receptor, and it binds there. The S2 subunit of this protein enables the fusion with the cell membrane and allows entry into the cell. The nucleocapsid protein is the most abundant protein, and it's highly immunogenic too. To just speak a word so that you don't lose track or you don't remember the structure at a later point, I like to make a mention here is when the virus has so many proteins, when the whole virus is given as a vaccine, it would synthesize it, it would make antibodies in the body of the host for all these proteins together. Therefore, even if the spike protein is mutating and you have a query whether or not this vaccine will protect you, it will not. You're rest assured that the other antibodies that you're making in the body against all these other proteins which are present in the whole virus vaccine will protect you to a larger extent than the vaccine which has only the spike protein being addressed. So I want you to remember this, and this is why the vaccine of Bharat Biotech is more useful, because they are using the whole virus and not only the spike protein. So even if the spike protein mutates and there is a low binding of the mutated spike protein in spite of giving the vaccine, these are the antibodies which are present in the body will enable you to fight the virus. Now, going to this diagnostic test, we've already done a test. So what are the, one is you do your RT-PCR, you do your antigen testing, then you do what is called a a chest CT, you do a CBP, and so many other things. I'll not give you a complete list of what all is done, but oftentimes we go by what is called the chest CT, or the this is not very accurate test, simply because chest CT will show you a damaged lung picture, even if you have TB, if you're a smoker, if you have other viruses, respiratory viruses that are impacting on the lung. It, I mean, if your RT-PCR is positive, then this chest CT is indicative of how much extent the lung is damaged. We are assuming at that point that the other causes for the chest CT are not all these other viruses or other conditions, but the damage is purely because of COVID. So this is a collaborative test rather than a diagnostic test. So the significant percentages of patients COVID have a normal chest X-ray or X-ray, but for those who with a normal imaging result could falsely convince them that they are healthy, but the test on which you should depend is the RT-PCR test. Now coming to the immunity and immune response. Okay, what happens as soon as the virus enters the body? The body knows that something foreign has come in. 1 minute. Okay. So the immune response, the two major cells that are important in the body, which are called the B cells and the T cells. These are kind of lymphocytes which are present in the blood. So what happens is the macrophage, which is a kind of cell that is present in the blood, first sees this virus or any pathogen that comes in contact. It goes around it, welcomes it, gyros it, and takes it inside the cell. And now imagine that this is the virus and this is the phagocyte. What it does, it puts its arm around this pathogen and then internalizes it into itself. Like, open the door and call your guest home. And once the guest has come here or the virus is inside the cell, this cell senses that there is a foreign, just as in a hotel room, once you go, you have the tag that says occupied, unoccupied. So similarly over here, this cell puts out a signal here that it is occupied. Now, what is the signal announcing the occupation? This is the antigen of this virus that has come in or any pathogen. This antigen is put on the membrane of the internal macrophage, and this now the T cells or the T helper cells, which are again floating in the blood, recognize and say, oh my God, there's someone who's come in here. This fellow has a pathogen that it is housing. So immediately it takes that address, and then this T helper cell now interacts with the B lymphocyte, which is again present over there, and then it says, hey look, ये कोई बाहर वाला आ गया है, चलो चलो इसको हम धूम मचाएंगे, or we'll kick him out. So let's build our body defense. So now this cell this T helper cell from here binds with the B lymphocyte, and this B lymphocyte internally synthesizes what we call antibodies. The first antibody to be delivered is IgM, and the second antibody that it has is IgD, and then you make IgG later on, and these two together hold all these foreign pathogens that come in, and then again take it in like this, and these are lysosomal material is poured on it, and then it is killed and removed from the system. So this is how These B cells that are there retain a memory. Remember in the earlier cell, I said that it has this thing internalized. This whole B cell makes a big colony, and this colony, just as we get our salary, we put our money partly into the bank, partly into using today, partly for using now, partly for old age. So this B cell now, this colony that has made this marker with this particular COVID virus now makes a whole lot of clones or a number of B cells. Some go into making antibody immediately to fight your infection, and some go in as memory cell. So maybe subsequently at some other time, maybe after a year, you're exposed to COVID again. Your body already has a memory of these B cells, and it doesn't have to go through all this process. It quickly makes the antibody IgG, and you go neutralizing the incoming virus. So that is what these memory B cells, whenever it's attacked by a virus, so far your B cell was naive. Naive means it's not seen the virus. So now when this B cell, the virus is coming from outside, this reminds it that, oh, this is COVID virus. And then this fellow makes a whole lot of antibodies here against COVID and then neutralizes this COVID virus, and your immune system has worked, and therefore you don't get go down with the COVID infection. Now, how does this happen? So I think when you're being vaccinated, everyone has said you take the first dose here and then you wait for 2 months and then only you take the second dose, all these kind of things. Why are they doing that? So what happens is the first 7 days when you're primarily challenged with it, there is a lag time for you to make antibodies. And the first time you make antibodies, this is IgM. And after some time, this goes down into a sleeping mode or a dormant mode, and it is only after 1 month to 35 days, or in fact 42 days is when your IgG peaks. So at this time when you give the booster dose over here, that is when it will make a large number of the memory B cells, and therefore you're given second vaccination after 40 days or after 2 months or at least 1.5 days. So if you wait for 2 months, then you have a good peak of the secondary response, and you will get a high titer of these memory cells so you can store it in your system. Okay, so this is again just showing the same thing again. Okay, now when we do a serological test, how does that impact us? And what is the clinical significance? How do we interpret each of these? Excuse me, 1 minute. So the quantitative PCR is done when the patient is in the window period of infection. Now, this is the window period of the infection. This is when your symptoms are ready. And here is where you can detect your antibodies, that is the IgM. And it is only from here, after 14 to 21 days is where you see a lot of IgG coming, and the IgG that you're producing remains for a lifetime in the blood. Probably the titer goes down, but invariably you'll find a few at least. And if there's a second infection here, immediately this will boost, and again it will pick up. Okay. So that is the window period. And then you have in the early stage of infection, you will only have RT PCR positive, IgM positive, and this IgG will be negative because you're not yet started making. But the patient may be in late, then IgM is not observed. it is IgG that is seen, and over time your RT-PCR becomes negative, and there might be a little IgM and no antibody. And so at the end of the day, there is no RNA or the test for RT-PCR is negative and your IgG is highly positive, and that is when you're convalescing. All right, this is the cycle which shows the same. This is the theoretical part, and this is a graphic representation of the same. Okay, so other tests, okay. Research is still progressing because we have so many confusions with the RT-PCR also. So these are some cheaper tests where R&D is going on, which is called LAMP-PCR. The LAMP-PCR can be done even at the clinician's table. You don't have to go to the lab. This is a more modified, cheaper method where research is going on. For though it's an established method for TB and other diseases, for HIV, for COVID, it's in progress. So now what happens in the sorry, what happens if I had to go through the life cycle with the events post exposure? What happens? You're exposed to an aerosol, and the aerosol has transmitted the viral infection to you. So this is now attached. Attachment occurs to the cells in the system. The S2 receptor and the S1 subunit interact, and the virus is attaching itself to the cell. Then there are conformational changes of the spike protein. It penetrates the host cell. Then it releases the virus. Viral particles are replicated. It matures to form a full virus, and these viruses are now released into the atmosphere, into your cough, into whatever else, and then it goes into the atmosphere and meets another person, and this whole cycle keeps going. So now coming to the disease physiology, what happens? Either you come down with the disease when you had a lot of viral particles entering your system, or one or two, nothing happens to you, you go away. So first and foremost is, nature is very, very nice with us in the sense that, we have what are called portals of entry. At the portals of entry, we have various methods by which we stop the foreigners. All right. So just as when you're going from one state to another state, your visa is checked, your baggage is checked, your physical check. There's so many different levels of checking. Similarly, over here, when a virus or any pathogen enters through the route of entry, and the first thing is, I don't know, your nostrils have very small hair. These are called cilia. So, I mean, I'll not go into the details of this unless you ask me in the question session. But then, when you enter, the virus enters the nose or throat. The very architecture of the tunnel over here to the lung is kind of circuitous. It's not direct like this, that it's like a charaha, wherein you have to look at the direction, which road will take you to Pune, which road will take you to Hyderabad, or will you go back on that same thing. So similarly over here, when the virus enters the system, at every point you're asked to wash your hands with a proper spirit and other things over there, wherein it's all ill, and you're asked not to touch your face, your hands, your eyes, your nose, everything. So every organ on your face, whether it is lips, whether it is your eyes, whether it's your ears, or whether your nose, there are innate immunity mechanisms which prevents the virus from coming. And if the virus load is very high, you gate crash, then it goes further down, and there's a check here in the throat. Further there's a gate crash, then it goes down the lung. So the viral particles then replicate, and there's a local propagation, and with a limited immune response. Then, when it goes further down to the airways and lower respiratory tract and invades your pulmonary alveolar tissue, there could be two options over here. One is that a fifth of the patient are very comfortable. Yahin pe, they are the incoming pathogen is knocked out and nothing happens to them. They're hale and healthy. Another one enters into a pathway which is, a moderate infection or a severe infection. And here, in the moderate infection, there is a certain amount of viral replication, but then every cell that is housing the virus, there is a process called apoptosis, wherein the body itself kills this infected cell, and therefore that dead cell is again removed by various mechanisms, and then there are adjacent. If it doesn't, the resulting viral particles infects the neighboring healthy cells, and the process of infection continues till it becomes acute respiratory distress syndrome. Okay, and or otherwise, right at this point you have a cytokine storm where so many molecules in the body are released, which is IL-1, IL-6, IL-8, IL-12, TNF alpha, interferon gamma, interferon beta. Probably you read the newspaper that now interferon is one of the treatment protocols which Zydus Cadila is bringing out. And incidentally, I worked with Zydus Cadila. I cloned the interferon gene for them, but it was shelved at that point in time. But today it has been revisiting. They have pegylated the interferon that I had cloned, and then they're going through a clinical study for using this as a therapy. So any one of these interleukin molecules which are storming the system, you can make antagonists for any of these to become an antiviral therapy. As of now, all of them do not have antagonistic molecule in the market, but sooner than later there shall be one. Then you have chemo attraction for neutrophils and the CD4, CD8 cells, which are all different kinds of lymphocytes which are present in the body, in the blood, which along with B cells differentiate, and they attempt to defend the host and clear the viral particle. But if besides all this, if there is acute respiratory syndrome, then they're put in intensive care and monitored well, and some are lucky to come out and some are unfortunate. Okay. So here we have three categories of the disease: mild disease, moderate disease, and severe disease. So I'm leaving the slide with the organizers. So this is something, for our, you're kept in isolation, and these are the kind of medication that you're given. This is in home isolation. This is in hospital isolation. This is in intensive care. So if you want me to quickly go through it, there are I've discussed about what are the home care. In the earlier slide I told you, like, one can be at home and take care, or they can be in hospital, and these are the schedules that are given. So self-care quarantine, I'll let you read this from, because I want to dwell more on the vaccine rather than these. Okay. So these are the approaches to control infection, community, individual. Okay. So one is to break the chain of infection. What we do, we had a lockdown. We are hoping that we'll have a herd immunity. Aside of all this, the main thing that we are doing is we want individual immunity, or every individual, we want them to control the infection. For this, using these 7 types or material. Either you can have a live attenuated vaccine, inactivated vaccine, a subunit vaccine, a toxoid, conjugated vaccine, recombinant vaccine, nucleic acid vaccine, etc. And all of these will give any one of these responses or a combination of these. Now, each of these vaccines I shall explain in detail now. Okay, so what are the participants in the game for vaccination? You have the offenders, you have the defenders. The offender is the virus, and the virus again is COVID. The defender here is the host, which is the human system. And in the human, you have the immune system, which is, it could be cell mediated or antibody mediated. And this is within the body. Now, the external is you give therapy, that is medication, or you give immune modulators like interferon, etc. Now, in terms of immunity, I was speaking to you a while ago that there is innate immunity, acquired immunity, and what we are speaking of. Okay. Innate immunity, I did not deal much, but we have anatomical barriers, which is what I told you. Your mouth is kind of, the saliva has acid in it. If it goes to the mouth, a little of it gets killed there. Then you have, in the nose, you have cilia, which again kills it. Then you have phagocytic barrier, the cytokines, etc. Then acquired immunity, you know, if you're constantly working, there is some amount of immunity that we develop. But what we are concerned about today is artificially acquired immunity, which is what we would call vaccination. So in the body, there are two sites of immune response. One is systemic and one is mucosal. Now, systemic is the whole body, wherein it comes in the blood circulation. Your blood carries the antibodies. The mucosal compartment is, if you look at all the soft tissue in the body, whether it's the digestive tract, the respiratory tract, the genital tract, or whatever, all these organs which are directly opening into the environment, like your mouth opens into the environment, your nose opens into the environment. So all these portions are covered with a protective layer called mucus. That is why, you know, your eyes, again, when something falls, immediately tears come out. So whatever is coming comes out with the tear. So the body has these kind of mechanisms over and above which we need to make antibodies. So there are these portions of the body which are covered with mucosal uh, cells. And in the systemic cells like spleen, liver, brain, etc., are the systemic compartments in the body, and these do not have mucous membrane, and therefore they are dependent on the blood or the systemic antibodies that you generate. So the first method of defense is the mucosal antibody or the mucosal components that are going to get you rid of the incoming pathogen. But nevertheless, as infection proceeds, today we do not have any mucosal vaccine ready. But if only we could make a mucosal vaccine, that would be the best vaccine that we could have, because it will be active at this very site. When the virus is entering through your nose or mouth, you're taking care of it then and there and not waiting for the disease to come up. Okay, so in humoral immunity, this is what I told you a while ago. The virus comes here, then, you know, this whole mechanism wherein the B lymphocytes, you make memory cells, and these B lymphocytes secrete a whole lot of antibodies, and these antibodies are going to neutralize your incoming virus. So it's the cell-mediated immunity. This is the B cell and this is the T cells. So I told you the T cells and the T helper cells are what going to label your B cells to make antibody making cells, and these will make a lot of antibodies. So there is a cooperation between the B cell and the T cell which makes antibodies. Okay, so when you're making a vaccine, the things that you need to bear in mind are: is it effective? Is it available? Is readily available? Is it stable? Is it affordable? Is it safe? So effective is, it must evoke protective levels of immunity at appropriate site, relevant nature, and adequate duration. Availability, is it can you make a huge quantity of it and distribute it to everyone who needs it? Is it stable under your climatic conditions? Is it affordable? Is it priced very high, and especially in the developing or underdeveloped countries, can you afford it? Is it safe? Apart from eliminating the pathogenicity, is it doing any other harm? Okay, so for this, there are various vaccine strategies. You're using either inactivated virus, which is what Bharat Biotech does, whether you're using attenuated virus, or whether you're using a subunit vaccine, or a virus like particle, or a DNA vaccine, or a viral vaccine. So these are different strategies that today we are using for vaccine preparation. So the types of vaccine, as I've already told there, you have whole virus, protein subunit, viral vector, or nucleic acid vaccines. So what is vaccine? Definition is here: are biological preparations made from living organisms to provide a prophylactic or a therapeutic action. So some of them smuggle the antigen into the body, and others use the body's own cells to make the viral antigen and make antibodies to combat infection. So the whole virus vaccine, there are two types over here where you're using the whole virus. One is live attenuated and inactivated vaccine. Here, the live attenuated, it is passaged in a particular way where it loses its pathogenicity. Inactivated virus, there are different inactivating agents, like it could be X-ray, it could be gamma rays, or it could be chemical inactivation, wherein the replicative or the nucleic acid of the virus is knocked off, and therefore even if it goes into the system, it cannot cause any infection. So, for example, over here, the indigenous inactivated, the vaccine that Bharat Biotech makes is an inactivated vaccine. The indigenous inactivated vaccine is developed and manufactured by Bharat Biotech. The whole virion is inactivated in viral cell derived platform technology, and this is what is administered to people. Now, cyanovac is from China, and they make it is a whole virus vaccine. It's an inactivated pathogen. So, if the inactivation is incomplete for whatever reason, maybe if you take 1 ml of the vaccine, and even if one or two viral particles are live in that sample, God forbid, but then if it does enter your system, that one or two viral particles can mount to thousands, millions of viral particles in no time at all, and the patient can come down with the disease. Now, nucleic acid vaccines, here it could be an RNA vaccine or a DNA vaccine. Um, So in this case of your, this is usually you use the RNA of the viral spike protein, not the complete viral RNA, but uh, only the uh, only the viral RNA which codes for the spike protein, or you take only the gene uh, necessary for making viral protein for making a vaccine. So once this genetic material gets into the human cells, it uses our cell's protein, because only this RNA of the spike protein is entering the human body. So the human body now makes a whole lot of spike protein, which the T cell and B cell see and make antibodies. So the advantage of such vaccines are they're easy to make. I don't have to make a huge amount of spike protein in the factory, because I'm going to make a small amount of RNA of the spike protein and inject into the host system. And the host, he or she, will make the spike protein, and the spike protein the T cells and B cells see and make their own uh, antibodies. So, since the antigen is produced inside our own cells and in large quantities, this is cheaper, it is safer, you don't have to sit and purify them, you don't have to bother about its stability, whether it's going in ultra cold temperatures or not, etc. We don't have to worry. But then the RNA itself is a very, very unstable molecule. That is why if the virus is present on the dust or somewhere, we are not very, just by wiping it, it is enough. It's taken care of because RNA is very, very fragile molecule. The life of an RNA, if it is naked, if it is not covered with the protein coat that it has, and if the viral RNA is lying naked anywhere, it takes only 30 seconds for it to die. But then, unfortunately, when the virus particles, I cough or spit, and it's lying on the table or a chair or the doorknob, it has the protein coat which is protecting it, and therefore its life is enhanced. Now, this viral vaccine again, because it's only RNA, it has it's maintained at minus 70 degrees, and in India it's very difficult for us to, this is the Moderna vaccine, wherein to maintain minus 70 degrees in transit is extremely difficult, and which adds to the cost of the virus, and therefore we do not have Moderna vaccine in our country. So the viral vector here again, um, in the viral vectors again, the vector itself carries. Now, what's a vector? I'll show you in another slide. I'll summarize all this in a diagrammatic manner in a little while. So, the viral vector vaccine also works by giving the cells the genetic instruction to produce antigens. That means it takes only the spike protein. All these, except the Bharat Biotech one, is addressing only the spike protein. And that is the only reason why I would prefer Bharat Biotech vaccine as against all of them. Because even if you were to believe that the spike protein is mutating so very often, then and the avidity or the avidity means the binding capability of the vaccine and the infectious agent may or may not be a strong fit. Sometimes, if it's a loose fit, its efficacy falls down. But then, if you're looking at the whole virus vaccine, it has all the other proteins, and all of the other proteins are not mutating at all. And therefore, the sum of the immunity is the sum of all these antibodies together as versus one. Maybe spike protein makes 100 copies. I'm just giving a rough number, 100 copies. Okay, and maybe the other proteins together make 200, but each of them make less than 100. All right, but the sum total of all this, even if the spike protein becomes 50 in terms of its efficacy, all these proteins together make more than 100 antibodies to fight the incoming virus. Okay, so in the viral vectors, like it could be adenoviral, I'll come to that. Today we are using adenoviral vectors. What is a vector? It's a small viral particle which is constructed in the laboratory with the traits we want it to have. So, you put functions, you make, it's like a bangle, it's like a bangle. If this is the whole vector, I put one side A, B, C, D, E, whatever. I designed this, putting all the characters that I so desire to have in this vector. But one of the main things I put here is the spike protein gene. I put something for the so that it can enter. It should have a property to enter the biological cell. It should have a property to go in there and make more spike protein. So I take each of these characters and I construct a viral vector. I am the scientist. Okay, this is what I did for hepatitis B at one point in time. They differ in the nucleic acid vaccines in that they use a harmless virus. So the virus used here is the adenovirus, which doesn't infect human beings but infects chimpanzees. Okay, different from one vaccine is targeting to deliver these instructions into the cell. Instructions for what? That it should go inside the cell, it should make take the spike protein along, and it should make more spike protein. So the one type of virus is adeno. I said that already. So this nucleic acid is hijacked by the instruction in the cell, and it makes more and more antibodies. So the viral vector vaccines are made. Zadis Cadila is making it, and Johnson and Johnson is also making it. So that's what it is. Now, DNA vaccines, instead of using RNA virus, you can use a DNA, which is a plasmid. Plasmid, all of you biology students must be knowing that there are many kinds of plasmids which are extranuclear organs that are present in bacteria. So you take this bacteria and you engineer it in a manner that it has characters of choice. Okay, so here again the DNA vaccine, the RNA from the virus is extracted. Remember, COVID is an RNA virus, so the RNA is extracted. This RNA is converted into DNA. This DNA, the genetic sequence derived generated from the DNA is amplified. The target gene over here will be your spike protein, and this is how a viral gene is there. You take out only the spike protein from here. This is a plasmid, and then you put the viral gene here, and this whole plasmid is injected into the human system. So when this is injected into the human system, I mean, this is only telling you, details is not required. Like, you need restriction sites to cut, stitch, whenever you're making a construct. कपड़े में आपको shirt या कुर्ता बनाना है, तो आपको size के माफिक, you have to cut the clothing, and then you have to stitch it, you have to lining it, all these kind of things. So similarly here, when you're doing a construct, you need a scissor to cut wherever you want, which is called, the biological scissors are called restriction endonucleases, because of which you cut wherever you want. All right, and then you have to join it. So instead of needles and thread to sew it over, we have enzymes called ligase that you sew it. And then again, you need markers to know that this is sewed in or not. So you put some kind of a tag, an address tag, to show that this portion is present completely, etc., etc., for each one of them. And you selectively grow. You have markers which will take only this DNA and not anything else. So you put something like that. When you go to the mall, you have size specified, right? 34 number, 35 number, 36 number, 22 number, 18 number, whatever. So like that, you have markers over here where you can select this against all else, and therefore you cut the gene, you take the gene, you put the gene, you grow them, and then you add an immunogen over here is a spike protein. You insert that into a plasmid, you transform bacteria and grow tons of it, then you extract it, and then you give it to the animal to do a safety study, and then once it's done, you give it to the human being. The RNA vaccines here again, I've told you this is the same method, but I'm giving you more details of the messenger RNA, which is the Moderna vaccine, which has encodes for the spike protein, which is a glycoprotein, and a transmembrane anchor, which I told you in the very first slide. Where are you cutting it? You're making two substitutions. I mean, it's all detail of the design. Like, I want a sleeveless, I want a spaghetti sleeve. I say, like, when you're designing these, you have different kinds of modalities that you fix in and do accordingly. All right, this is how it is. So this is a SARS-CoV virus, this is a spike protein, and you take the spike protein, this is a complete genome, and this is the spike protein region. You take the spike protein, separate it, get it, and then you construct a whole plasmid, and then you vaccinate it, and it goes into the body, into the cell nucleus, and this makes more and more of this spike protein, and the spike protein is all over your system. The B cell sees it and makes so many antibodies for the spike protein, and when the virus from the environment comes, these IgG molecules are ready to capture all these and send them out of the system. All right. So this is how the corona RNA virus vaccine works. This is a coronavirus. You're taking the RNA, it goes into the cell, the spike protein, and now the cell, T cell, B cell, then it makes the antibody, and that's it. Now, protein vaccines. So, in protein vaccines is made by a company called Clover Pharmaceuticals. They make SARS spike protein, which is similar to RNA virus, such as HIV or influenza or whatever. They make a trimeric spike protein on its viral envelope, and this spike protein, along with an inactivated or dummy virus, is injected into the body. Okay, this is how. So, contains SARS-CoV-2 virus, which is recognized with the immune system. The whole protein and protein fragments or many protein molecules are packaged into a nanoparticle. So all these are packaged into what are called nanoparticles, which could be a biological nanoparticle or it could be a chemical nanoparticle. So these nanoparticles are now injected into the body and they make antibodies. This has been successful for Hep B, but as of now, the clinical trials for COVID nanoparticles is being done by Novavax, Sanofi, GSK. Okay. Now the major international companies are Pfizer makes mRNA vaccine, Johnson and Johnson makes viral vector, Moderna makes messenger RNA, AstraZeneca makes adenoviral, GlaxoSmith makes this, etc., etc. In the Indian scenario, we have Bharat Biotech making the Covaxin, Serum making AstraZeneca Covishield, Zydus making molecular DNA, and these people are also in the race. So now, whenever you're making a vaccine or anything, you need to have regulatory approvals. Now, what are these regulatory approvals? It has clinical phase 0, phase 1, phase 2, phase 3 trials, and only then it can be given to humans. So, in phase 0, you're exploring if and how a new drug may work, often with fewer than 15 people, and the drug is given only for a short time. In phase 1, you're ensuring the safety treatment, whether it is safe or not. About 15 people are given low to high dose, like 15 people are low dose, and there is a gradation of the dose. And in each group, you have 15 healthy individuals taking these medications and seeing how it does. In the phase 2 clinical trials, you take if it is safe in 1 and 2, then you go to phase 2 where you're taking 100 persons in different groups and 25 to 100 people in different doses, and then you randomize and give it. Then in phase 3 clinical trials, it's better than what is already. It's a randomized double blind. That means you have one group getting the treatment and the other group getting, maybe we call it placebo. It could be glucose water, it could be a tablet, whatever form we are giving to the other group. The form will be the same, but it will not have the active ingredient. But who gets what, it is all barcoded, so we do not play with the data. So the adverse, all vaccines may have some amount of adverse effects. Nothing is 100% correct because we are not God. However much we claim to play God, we are not really God. And therefore, there would be some amount of side effects. The common side effects is there will be a slight warmth wherever you've taken the shot. There'll be a slight pain, a little swelling for a couple of days, and you rest your body. You may have fever, chills, tiredness, headache. These are the common ones. But if at all there's anything serious, then you should speak to your clinician. That depends on what your comorbid conditions are. If I'm already a heart patient and I'm on certain other medications which reacts with this, that is why it's very important that you share all your health data and not hide from the clinician who's attending you. So this is again a summary, like who's doing what and which is present in the market. Okay. So what is the lacuna and the future possibility? The major vaccines available in the market are all for systemic use and none for the mucosal. And but corona is a respiratory infection, and therefore, if we have a mucosal vaccine, that would be the best. So the two important regions of the body I've already said, systemic and mucosal. So the mucosal vaccine is best for respiratory viruses, but as of today, why are we doing systemic first and not mucosal? Because the mucosal vaccine has certain fundamental uh, what do you say, speed breakers for making the vaccine. All these mucosal cells are very rich with a teeming amount of hordes of common cells or friendly microorganisms present there. So if you're doing some therapy target, then this interferes and we have to do a lot more to get rid of them or make screens possible for testing the incoming mucosal vaccine and remove all these mucosal vaccines, and that becomes a critical issue. But it is present, like we have done it successfully for polio, we've done it successfully for some of the pox infections, and therefore it's not impossible, but it takes a little longer time, and therefore our first attempt is to do a systemic infection, and uh, we've gone ahead and so many vaccines in such a short duration. Though the virus epidemic itself or the pandemic started, let's say in January of 2020, by January of 21 we have so many vaccines in the market and it is being protected. So if we have a mucosal vaccine, it also stops shedding of the virus. That is how we were able to contain polio outbreaks. Okay, so IgG cannot reach the mucosal layers, and only the mucosal vaccine, which is IgA, will reach the mucosal cells and cause a antigen antibody reaction, neutralizing the incoming pathogen. So this is a kind of summary where it's just, thought processes. There's nothing to really explain over here. But then, you know, um, you can read this, and any doubts you have, you can come back to me. Okay. So to summarize, we have AstraZeneca, Moderna, Zydus Cadila, Bharat Biotech, which are all in the market today. Thank you for your time and patience. And now maybe I will